Video's set of questions is related to cell biology. Let's begin. First question is, in which cell organelle nucleic acid is not present? Chloroplast, mitochondria, Golgi apparatus और राइबोसोम. क्लोरोप्लास्ट में उनका न्यूक्लिक एसिड होता है, माइटोकॉन्ड्रिया में न्यूक्लिक एसिड होता है. दे डू देयर ओन डीएनए रेप्लिकेशन. गोल्गी apparatus में न्यूक्लिक एसिड नहीं होता है. राइबोसोम जो कि राइबोजाइम से बना है, उनके पास भी न्यूक्लिक एसिड होता है. सो द आंसर विल बी गोल्गी apparatus. It is the cell organelle that doesn't have nucleic acid of its own. Next is, which organelle help in the synthesis of lipids, cholesterol, steroids and visual pigments in epithelial cells of retina? कौन से ऑर्गेनल हैं जो लिपिड का सिंथेसिस कराते हैं, कोलेस्ट्रॉल, स्टेरॉइड्स का सिंथेसिस कराते हैं? वो आंसर इज SER, that is smooth endoplasmic reticulum. Next is, elastic nature of plasma membrane is due to the presence of. जो प्लाज्मा मेम्ब्रेन के इलास्टिक नेचर होता है, फ्लूइड नेचर, उसको कौन देता है ऐसे? प्रेजेंस ऑफ लिपिड्स के वजह से वो इलास्टिक नेचर का होता है. तो आंसर विल बी ए, लिपिड्स. नेक्स्ट इज, which cell structure substructure act as the site of oxidation reduction reaction? कौन से सेल के सबस्ट्रक्चर में ऑक्सीडेशन रिडक्शन रिएक्शन होता है? ग्लाइऑक्सिसोम्स, माइटोकॉन्ड्रियल क्रिस्टी, lysosomes, coated vesicles in Golgi complex. Answer will be B, mitochondrial cristae. Next question is, middle lamella of cell wall is mainly composed of hemicellulose, protein, carbohydrate or calcium pectate. Cell wall middle lamella, calcium pectate is present. So the answer will be D, calcium pectate. Next is, which organelle serves as site of modification, packaging and preparation for the distribution of newly formed proteins? So the newly formed proteins are formed in the cytoplasm, and the organelle responsible for its site of modification and packaging preparation is A, Golgi complex. The next question is, membrane covering vacuoles in plant cell is टोनोप्लाज्म, सेल मेम्ब्रेन, सेल वॉल और टोनोप्लास्ट. तो वैक्यूल जो है वो प्लांट सेल में ही प्रेजेंट होता है सिर्फ. इट इज स्पेसिफिक टू प्लांट सेल. एंड द मेम्ब्रेन कवरिंग वैक्यूल्स इज actually termed as tonoplast. Okay, so answer will be D. The numbering is incorrect here. The answer is D. At which phase size of chromosome is measured? कौन से फेज में हम साइज को मेजर कर सकते हैं क्रोमोसोम का? The answer is मेटाफेज. इसमें हमें अच्छे से क्रोमोसोम विजिबल होता है. Next is, cell theory states all cell have nuclei, all cells are living, cell reproduces by both mitosis and meiosis, cell are fundamental structural unit of plants and animals. So there is some mistake in the question. The question will be actually, the cell theory doesn't state. इनमें से कौन सा स्टेटमेंट गलत है according to cell theory? So the answer is actually D, that is cells are fundamental structural units of plants and animals. Also because bacterial cells or other microscopic cells are also there. So the answer will be D. Next question, analyze the molecule that helps to add stability to the bilayer of plasma membrane. कौन सी हमारी मॉलिक्यूल होती है जो स्टेबिलिटी देती है, रिजिडिटी देती है हमारे बाइलेयर? बाइलेयर में क्या प्रेजेंट होता है? हमारा फॉस्फोलिपिड्स, और उसको हमारा स्टेबिलिटी देती है कोलेस्ट्रॉल. आंसर है बी. नेक्स्ट क्वेश्चन, मेम्ब्रेन कवरिंग, सॉरी द क्वेश्चन हैज बीन रिपीटेड, we'll skip this question. These both questions have been repeated. Next question is, lysosomes are rich reservoir of. Lysosomes हमारा क्या कहलाती है? It is the other name of lysosomes are suicidal bags. It contains some enzymes called hydrolases. So answer is A. Hydrolases help to degrade all the molecules that comes to lysosome for degradation. Next is, secondary structure of integral protein that is buried in lipid bilayer of cell membrane is. Secondary structure of integral protein, there are 2-3 types of proteins that are present in the bilayer, lipid bilayer: integral protein, peripheral protein. Basically integral protein is transmembrane protein, and which the secondary structure of that protein is of alpha helix. Also there is An exception, uh, a protein, integral protein called porins present have beta strands. Here the answer is alpha helix. Next question is organelle responsible for the origin of lysosome is. So the lysosome is originated from the Golgi bodies. Answer is C. The next question is plasma membrane is composed of proteins, lipids, cellulose, both A and B. Plasma membrane are composed of proteins and lipids both. So answer will be A and B, and cellulose is present in cell walls. Next question is cell theory is not applicable to. Cell theory is actually not applicable to viruses, as viruses D. Next question is cell recognition and adhesion are facilitated by the components of plasma membrane. These components are generally we have to say the nature of the components. Protein molecules alone, lipid molecules alone, both A and B, or glycolipids and glycoproteins. So the carbohydrate structures like glycolipids or glycoproteins give the property of cell recognition and adhesion to the plasma membrane. The answer will be D. Next question, organelle having ability of detoxification. Cell organelle having ability of detoxification is which one? Smooth endoplasmic reticulum, ribosome, Golgi body, or chloroplast. The answer will be SER, that is smooth endoplasmic reticulum. And the organ having the ability of detoxification will be liver gland. According to fluid mosaic model of plasma membrane is composed of which of the molecules? Phospholipid, intrinsic and extrinsic proteins, lipids and fats, cellulose and hemicellulose, both A and B. So here the answer will be phospholipids, intrinsic and extrinsic proteins like integral proteins or peripheral proteins. The answer will be A.